Hi, my name is Steve Taylor. I'm Chief Scientific Officer of Zegami, and today I'm going to talk to you a bit about our predicting heart disease collection. So, coronary heart disease is the leading cause of mortality in the world today. It accounts for about 7 million deaths a year. 110,000 of those are in the UK, 366,000 of those are in the US, and you can use echocardiogram as a diagnostic for heart and also a range of other conditions. And the good thing about doing it in a more automated way is you reduce the amount of time, obviously, that a physician would have to look at the data. But the other thing is it's more reproducible and less subjective to particular experiences of the operator. So just to put this into context, here we have a picture of a heart which has been made semi-quantitatively, and the method you can find out here in this paper, which is an IEEE paper back in 2018 about how to make these what's called so-called bullseye maps. But in a nutshell, what you do, you take the echocardiogram data and you mark out where the particular regions of interest here are. So this is the bullseye region here, which is sort of looking down at the top of one of the atria of the heart, and then you measure the blood flow in it, which you can do semi-quantitatively. So you've got this region here, so you can see very dark is very poor blood flow, very sort of yellow is high blood flow. And then we can use some simple statistics in Zegami to separate out the difference between somebody who's got healthy disease, somebody who's healthy, and somebody who's got chronic arterial disease. So here we have 127 patients that have had this procedure done to them, and actually in this case we do know which one have normal echocardiograms, and we know which ones have got more diseased echocardiograms. So you can see here, obviously we can zoom in at every level. We can take an overview of the amount of data we've got. We've got this view here called graph view, and we can look at this. We can look at this sort of numbers of disease items here. You can see we've got roughly the same 62 and 65 in the chronic arterial disease here, and we've got 65 here. And to all intents and purposes, casually, you can see that there's a sort of subtle difference when you're looking at it by eye, but it's quite difficult to resolve. But what we can do, we can take these data and process this. So in a simple way, what we've done here is we've basically broken this down into a 100 by 100 grid, taking the average pixel value here, and then subjecting that to a dimensionality reduction technique called principal components analysis. And using that is very powerful in Zegami. So let's have a look at that. So let's just look at these first two components here that we've defined. So you can see that there's a pretty clear clustering effect going on here. And we can really look at these differences really nicely if we overlay the colour which shows the disease. So you can see here there's this clear delineation of control versus the group with chronic arterial disease. And we can, if we want to inspect these, we can turn that colour opacity down and be able to go and zoom in which ones are the margins. And you can see here there's obviously there's quite a lot of sort of like blood flow here at the bottom in the more sort of like control ones, but you can see around here where you do have the chronic arterial disease, even though there's quite a lot of blood flow in that area, it's definitely a lot darker. So again, just by scrutinising the data and just looking at it in a different view, you can really get a lot of extra value from it.